As you know, we have a practical on Thursday, okay? The way the practicals work is on the back bench I'll have a series of stations. Each one of those stations will have something you have to look at, okay? It may be a microscope, it may be a petri dish, it may be something that we've done experimentally, and then there will be a series of questions that you have to answer about each thing that you see, okay? Please keep in mind that the questions associated with each of the stations have to deal with the station, or what's in front of you, okay? I know it sounds kind of stupid for me to say that, but you'd be surprised how many people just come up with things out of left field and don't even look at what's in front of them, okay? So please make sure you do that. As far as the microscopes are concerned, I will have the staining and everything done for you. It will be focused. Your job is just to look at them, okay? With that said, I don't want you moving things around, other than the fine focus knob. It's a little blurry for you, okay? That's the only thing. There also will be some short answer and one essay question, okay? And those don't necessarily require the back stations, so you can sit down and answer those at your bench, okay? Any questions on how it's set up? All right, good. So let's go ahead and go into the review. So what I do for the first review is we're going to quickly go over something I created called Microscopy Review. This will give you some real-life examples and pictures of what to expect when you look under the microscope, okay? Then we'll talk about Chapter 3. As I said before, Chapter 3 is on the practical, not in the lecture material. Then we'll go through the lab manual, and I'll pick out some important questions that I think are important for you to know for the exam. And then lastly, I'll go through some of the practice problems for the CFU per mil calculations, okay? Make sure everyone's on the same page with those, okay? So let's start off with the microscopy review. This is easily, I was like, what is that? This is easy to get. If you go on Blackboard under the lab tab, you will find them, okay? I definitely recommend getting it. So the first slide shows shapes. You will need to know the different shapes of bacteria, and there are three primary ones: cocci, bacilli, and spirillum. Cocci are round, bacilli are rod-shaped, and spirillum are S-shaped, okay, or spiral-shaped. And then next to it, you'll see some real-life examples of what these look like under the microscope. Okay, so you get an idea of what you may see on the day of the practical. You will also need to know these four major arrangements: Diplo, Staphylo, Strepto, and Tetrad. So Diplo, you can see here, it's a little hard to see from far away, but if you look closely, you can actually see these little blobs here are two bacterial organisms stuck together. Okay? They look like little coffee groups. Next, we have Staphylo. These are grape-like clusters, so it looks very irregular. Strepto, which are linear, or chains. And then Tetrad are four guys stuck together. And down below, we have just a little cartoon diagram of what these look like, okay? So again, this is something that I may ask you on the exam, so make sure you understand what to expect from the microscope's perspective, point of view. Next, gram staining. So gram staining, as we've talked about, is a very important technique. It's one that gets used a lot in microbiology, and it's usually the starting point, okay, the first thing that we do. So with gram staining, not only do I expect you to understand what gram staining looks like under the microscope, I also expect you to understand for the exam how to perform a gram stain and what is actually happening during each step of the gram stain. Okay? So first off, what is a gram stain? What are we doing with a gram stain? What's the point? Does anyone remember?
Find out which ones that have capsules that
So ones that have the envelope that are thicker and only have 2 layers compared to the thinner and only 3 layers. Okay? So the thicker guys with 2 layers are going to be considered gram-positive organisms. The guys with the thinner cell wall and the 3 layers are considered gram-negative organisms. Okay? So don't worry so much about, taking your bacteria, mixing it with water, drying it, and fixing it. You don't really have to talk too much about that because that's the same for every staining technique. Okay? What's important for you to understand for the exam is starting with the crystal violet stage. So crystal violet, the dye, right? It's purple in color, and we add that to our organism. What's happening when we add that to our organism? So it's attaching to the organism, and more specifically, it's binding to peptidoglycan, okay, or the cell wall. So at this stage, everyone is purple because everyone has peptidoglycan. We then follow with iodine. What does the iodine do? Does anyone remember? It cements it to it, right? It creates a covalent bond between the crystal violet and the peptidoglycan. We call this a mordant. Okay? This makes it harder to wash away the crystal violet. Then we go into the decolorization. This is where we use acetone alcohol and we drip it on top of our organism. What's happening at this stage?
It's rinsing off the purple violet off of whoever doesn't.
So it's removing the crystal violet from the peptidoglycan. And the key here is that it's doing it only to a very thin layer of peptidoglycan. Okay? So because the gram-negative organisms only have a thin layer, they become clear. But the gram-positives have a thick layer, so they remain purple. Okay? Because you're only taking off the top, the rest of it remains. Now, we then follow with something called safranin, and this will dye the clear gram-negatives a red color. Okay? This is important because remember, they're clear, translucent, hard to see. So if you don't have the safranin, you won't be able to see them at least easily. Okay? Now, on the practical, this is an example of what you might see. Okay? For any of the differential stains, I will always have a mixture. So in this case, it would be a mixture of gram-positives and gram-negative organisms. Why do I do this? Well, usually the very first question I ask you is, what type of staining procedure is this? And you should be able to tell me it's a gram stain because you have purple and pink organisms. Okay? Now, I will do these organisms at different ratios. So like 90% one, 10% the other. If there are any specific questions about the organism you see in front of you, it's going to be about the majority. Ideally, I would like to have little arrows that I can point at things, but these scopes don't have those. Okay? So I have to just go this route. So that's the only one you need to know the protocol on. Okay? So we didn't do a capsule stain yet. We'll get to that later. But we did do an endospore stain, okay, last week. Endospore stains, remember, dye vegetative organisms red and endospores green. So if you were to see this under the microscope, you would know immediately that is an endospore stain because you've got blue, or excuse me, you've got green and you have red. Okay, green and red compared to purple and red. Another way to tell that this is an endospore stain is if you gently focus up and down with the fine focus knobs, what you'll notice is that the green stain will quickly disappear. And that's because it only reaches the very top layer of the endospore, so when you focus down into it, it will go away. It's another telltale sign that you're dealing with an endospore. And we haven't done those guys yet, or we haven't done flagellal arrangements yet, so perfect. So that's the microscopy review. Now let's go into chapter 3 quickly. Okay. So chapter 3, as I said, would be on the practical, and that's true. The good news is most of these things you guys have done in lab, so hopefully it's become a little bit more clear as the semester has gone on. So your book likes to talk about the 5 I's: inoculation, incubation, isolation, inspection, and identification. Make sure you have a definition of what each one of these are. Okay? So inoculation is taking your organism and putting it into nutrient-rich material, okay, and doing it aseptically, or not bringing in other organisms from the environment. Incubation is growing this now inoculated media at ideal temperature and atmospheric conditions for your organism, okay, so that it can grow and divide. Isolation, this is where you isolate your organism so that you can get rid of any other bacteria that aren't important to you. Okay? Anyone remember one way that we can do an isolation? There's 2 ways that we've done so far. Streak plate, right, so streaking it out. In this case, we're taking our organism, we're diluting it by spreading it over a large surface area. Okay? The beauty of this is that it's very quick. The drawback is that it's not quantitative, so you cannot determine how many organisms you started off with. If we want to determine how many organisms we started off with, what approach would we use then? Yep, the serial dilution method, okay, or loop dilution method. This is where we systematically dilute our organism, okay, in known quantities, so we can then back calculate and figure out how many we started off with. Good. Inspection, this is where we learn as much about our organism as possible. Okay? So we do a bunch of biochemical tests to understand what our organism is doing. Okay? And then identification is usually the end of the day, and this is where we take all of the information we learn from the inspection and figure out what organism we're dealing with. Okay? And remember, when we say pure culture, this means you have all of the same organism. Okay, so just one species within your culture. A mixed culture is where you have multiple different species, but you are expecting this. Okay? A contaminated culture is when you have multiple different species, but you're not expecting this. Okay? You were expecting it to be pure, but it ended up having more than one. And remember that colonies are single bacterium that have grown and divided many, many, many times. Okay? So there shouldn't be much variation within an individual colony. They are essentially identical clones of the original bacterium. So streak plate, as we mentioned before, quick and dirty. This gets you your end result very quickly, okay, in a couple seconds. But it is not quantitative. Loop dilution takes a little bit longer, but you can actually quantitate how many organisms you started off with, or what we call CFUs per mil. Does anyone remember what a CFU per mil is? Or CFU? Might be a better way to phrase it. Colony forming unit. All that means is a single bacterium that can divide and grow, okay, and form a colony. So essentially a living bacteria. Good. All right, so medias, there are 3 different types: liquid, semi-solid, and solid. Does anyone remember why we would use a liquid media? To catch gases, okay, like we did in the carbohydrate fermentation test. Or to grow very large quantities of organisms, okay, because you can incorporate oxygen in, it makes them grow faster. What about a semi-solid media? Locomotion, right? Looking for organisms and how they move. And then a solid media, why would we use this?
Isolation.
Isolation, right. So this is how we can actually isolate out our single organism. And remember, as far as nutrients is concerned, be either defined or complex. Defined means we add in every little bit and piece, exactly. Okay, it's artificial. Where complex is where we just take natural organic stuff and chew it up and stick it in the media. Which one's usually better for growth? No. Complex, good. It's better for growth because it's natural. Okay? We're taking a brain and chewing it up and putting it in there, and that's something the organism likes, right? Because it's naturally acquired. Defined is artificial, and so it tends to be a little harder for the organism to grow in. And then we can also define medias based on whether or not they can tell us something about the organism. Okay? And there's 2 groups, selective and differential. Selective is where you select for a specific organism. Okay? There are selective medias that select for either gram-positives or gram-negatives. And this can help you whittle down your focus a little. Differential medias, this is where everyone grows, but they change color based on biochemical characteristics. Okay? So an example of both a differential and a selective media would be the mannitol salt agar plate that we use, okay, MSA. The high salt means only gram-positives grow, gram-negatives won't. And if they grow and change the media color from a pink to a yellow, it tells you they ferment mannitol. So that is the selective side of it. Okay? All right, so don't worry about the basics of a microscope. I'm not going to test you on that. All right, on to staining. Fixing and staining. Why do we stain microorganisms? What is the benefit?
If we stain certain ones, certain nutrients will attach those bacteria, so then we'll know which ones they are.
Sure, yeah, we can stain so we can tell something about it, right? So we can do a differential stain and it tells us how thick their peptidoglycan is, for example. And also, by staining or adding a color to the organism, it makes it easier to see. Okay? It makes them pop on the slide, and then we can visualize them. Otherwise, it'd be very difficult to see. Okay? But the biggest drawback to staining is that you kill the organism. All of these dyes are deadly to the microorganism, and they will die. Okay? And remember, we can have a positive stain or a negative stain. Does anyone remember what a positive stain is? So positive staining is where you dye the organism directly. Okay? So gram stain is a positive stain. It's binding to the organism and dyeing it directly. A negative stain is where you leave the organism alone and you dye the background. You're still increasing the contrast because it pops from the background, but you are not dyeing the organism directly. Now, simple stains are the easiest. This is where we dye everyone the same color. Okay? Again, practically, this isn't done too often because it's kind of a waste of time. But when you're first learning, it's a good place to start. Okay? Keep in mind that when I have a microscopes in the back, if you look at an organism and you see everyone is the exact same color, what kind of staining would you assume that is? A simple stain. Okay? Remember, if it's a differential stain, I will have both organisms in the field of view, and you will see two different colors. So please keep that in mind. And again, differential stains dye different parts of the organism different colors, okay? Or different organisms different colors based on their composition. The most commonly used one is a gram stain, where gram positives are purple, gram negatives are pink. You can do acid-fast, which stains mycolic acid, and endospore, which looks for the endospore, as we talked about earlier. Okay, that's it for chapter 3. Now what I do is go through the lab manual, and I'm going to tell you questions that I think are important. Okay? So this will help whittle down the ones that aren't that important for the practical. This is a good time, if there's a question in here that you don't understand, to ask me about it. Okay, I don't mind answering. But these are the ones that are going to be the most important for the practical. So we'll start off on page 9. This is lab 2. Lab 1, remember we skipped. So on page 9, we've got numbers 1 and number 3. Okay? These are going to be the two most important on that page. Then we'll go to page 13 and number 1. Then to the next lab, page 18. Numbers 1 and number 2. And a lot of this is kind of redundant information from what we just talked about in the lecture. Okay? And then to the next lab, which goes to page 26. Numbers 1 and 2. And then page 30, numbers 1 and 5. Then on to the simple stains lab, page 33. Numbers 1 and 4. And then we skipped microscopy, so we'll go away from that one. Then we'll go to the gram stain lab, which starts on page 54 for the questions. Numbers 1, 2, 3, and 4. Alrighty. Then on to lab 7, microbial growth, starting with page 68. Numbers 1, 2, 6, and 7. 1, 2, 6, and 7. And then, nope, I scribbled that one out. Okay. Then on to the next page, or next lab, excuse me, pour plate. And that begins on page 81. So for this one, there is that formula on page 80. Make sure you know that. I won't give it to you. Okay. And then for the questions, it's 1, 4, and 5. And then there are the sample problems, which we'll go go over in a bit. And then page 86, number 1. Then on to error tolerance lab, which starts on page 92. And that's number 1, 3, 6, and 7. And then on 94, number 11, 12, and 13. And then page 101, number 2. And now today's lab, so resistance lab, this is the last one.
Did you repeat the last one, I'm sorry?
Oh, the last one was page 101, number 2. And then on to the resistance lab, so page 107, number 1, and then on page 108, number 7 and 9. All right, that's it for that. Now what I want to do is just quickly go through the colony count problems just to make sure everyone understands the math and how to set these things up, okay? So, we'll start off with number 1. Number 1 says 1 ml of water sample was placed in a petri dish with melted agar. After incubation, 82 colonies were counted. How many bacteria or CFUs per ml are in the sample? If there is no mention of a dilution, you can assume that the sample is undiluted. Use a 1 for the dilution factor.
What page are you on?
This is page 84. So this one's relatively easy. In the old lab manual, they didn't give you that last sentence, so it used to confuse the hell out of people. But really all this is your dilution factor. Oh, here, we can write the equation out just in case you guys don't remember. So CFUs per mil equals your dilution factor times your number of colonies over the amount plated. And this is always going to be in milliliters because we are calculating CFUs per mil. Okay? So, this one's relatively simple. You have a dilution factor of 1. You've got 82 colonies, and you plated 1 mil. That equals 82 CFUs per mil. Notice that I'm writing CFUs per mil after the number. That's very important in science. We never have just numbers, there's always something after it, okay? So we know what you're talking about. All right, on to number 2. 1 ml of milk was added to 9 ml of sterile saline. 0.1 ml of the diluted sample was put into a petri dish with melted agar. How many CFUs per ml in the milk if 200 colonies were counted? So we're starting off with 1 ml of our milk. We're diluting that into 9 ml. 9 plus 1 gives us a total of 10 milliliters. Okay? Because you're putting 1 ml into 9. Now I'm writing this as a fraction because it's easier for me. Our dilution factor is the 10. Whenever there's a 1 in the fraction, you just go with the denominator in this. We then can put that into our equation as our dilution factor. We counted 200 colonies, and we plated 0.1 milliliters. Okay? So we've got 2,000 over 0.1, which is 20,000 CFUs. Number 3. 1 ml of urine, it should say urine, not blood, is added to 99 ml of saline. If 0.01 ml were added into a petri dish with melted agar, how many CFUs per ml are in the urine if 40 colonies were added? So we've got 1 ml going into 99, gives us a total of 100. So our dilution factor is 100. We've counted 40 colonies, and we plated 0.01 milliliters. So we've got 4,000, 0.01, which equals 400,000 CFUs per mil. And if you want to be really adventurous, you can write this in scientific notation. which would look something like this. Number 4. 5 mils of pond water is added to 20 mils of saline. What is the dilution? So we've got 5 mils of pond water. We're adding it to 20. It's a total of 25. Okay. You can divide these two numbers to get the dilution factor, or you can simplify the fraction. Our dilution factor is 5. Number 5. 6 mils of urine is added to 30 mils of water. 0.1 mil is plated, 109 columns form. What is the CFU per mil of the urine culture? So we've got 6 mils going into 30, which is 36. Again, if we simplify this, this is a 1 in 6, or 6 dilution. 6 times 109 colonies over 0.1 mils. Anyone know what 6 times 109 is? It's above my mental capabilities.
65,000.
So 654? 6,540 CFUs. Number 6. 0.1 mil of a blood sample is diluted into 9.9 mils. 0.1 mil of this is then diluted into 9.9 mils. 1 mil is plated, 24 colonies form. What is the CFU per mil in this blood sample? Okay? So if we write this out as a fraction, it would be this. Okay? Because we're taking 0.1 mil, diluting it into 9.9 mils, which gives us 10 total. Now in fraction land, having a decimal is really bad to have in a fraction, okay? So we have to get rid of that. To do that, we just multiply both sides by 10. So that gives us a 1 in 100 dilution. Now we're not done yet. We do this one more time with this diluted sample. And so what that means is we multiply the two dilutions together. Because we're taking a dilution and diluting it further. Okay, and it's the same dilution, 0.1 mil into 9.9 mils, or a 1 in 100. We multiply these together, that is a 1 in 10,000 dilution. Okay. So our dilution factor is 10,000. The amount of colonies we counted are 24. And we plated 1 milliliter. 24 with 1, 2, 3, 4 zeros after it. Or 2.4 times 10 to the 5 CFUs per mil. Last but not least, number 7. 1 mil of sewage water is added to 9 mils of water. 0.1 mil of this is then added to 9.9 mils of water. 1 mil is plated, 243 colonies form. What is the CFU per mil of the sewage water? So our first solution is a 1 in 10. Our next solution is the same as before, the 0.1 into 10, which when we multiply by 10 gives us a 1 in 100. So to get the total dilution, it's 1 in 10 times 1 in 100. This gives us a 1 in 1,000 dilution. So dilution factor is 1,000, multiply that by 243, and then divide that by 1 milliliter. That's 243, 1, 2, 3 CFUs per mil, or 2.43 times 10 to the 5th CFUs per mil. That makes sense to everyone? Bringing out the old school arithmetic? All right. So hopefully you can guess that there will be at least one of these on the exam.
Some dilution 5 times.
Huh?
Don't do diluted 5 times.
No, I'll make the math easy.
Oh, okay.
I don't expect you to have a calculator. So any questions about the practical, what to expect, anything like that? All right, well, let's go on down to lab then.